A Carnegie Institution for Science audio press release for November 10, 2014. Biochemistry detective work, Algae at Night. Photosynthesis is probably the most well-known aspect of plant biochemistry. It enables plants, algae, and select bacteria to transform the energy from sunlight during the daytime into chemical energy in the form of sugars and starches, as well as oils and proteins. It involves taking in carbon dioxide from the air and releasing oxygen derived from water molecules. Photosynthetic organisms undergo other types of biochemical reactions at night when they generate energy by breaking down those sugars and starches that were stored during the day. Cells often face low oxygen conditions at night when there's no photosynthesis releasing oxygen into the air, and all photosynthetic and non-photosynthetic organisms in the environment are respiring oxygen. When this happens, some organisms, such as the single-cell alga Chlamydomonas, are able to generate cellular energy from the breakdown of sugars without taking up oxygen. They do this using a variety of fermentation pathways similar to those used by yeast to create alcohol. Although critical to the survival of common aquatic and terrestrial organisms that are found all over the planet, many of the details regarding this low oxygen energy creation process are poorly understood. There's new work from a team including Carnegie's Wing-Kang Yang and Arthur Grossman in collaboration with Matt Pozowitz at the Colorado School of Mines. The research hones in on the biochemical pathways underlying the special flexibility of Chlamydomonas in responding to oxygen-free and low-oxygen conditions. Other Carnegie co-authors include Claudia Catalanati, Tyler Whitkop, Luke McKinder, and Martin Jonicus. It's published by the journal The Plant Cell. In an arduous and exacting step-by-step process, the team used a series of specially created mutants to determine the importance of two identical branches of the fermentation pathway that are located in different compartments in the cell. They're both believed to be essential to dark, low-oxygen fermentation in Chlamydomonas. The pathways are dependent on four proteins, PAT1 and PAT2, and ACK1 and ACK2. ACK1 and PAT2 are located in a part of the plant cell called the chloroplast, which is the compartment where photosynthesis takes place. ACK2 and PAT1 are located in the mitochondria, the organelle in plant and animal cells where sugar breakdown takes place. Grossman says it was surprising when they found that the chloroplast pathway is much more critical than the mitochondrial pathway for sustaining fermentation metabolism. This was even though generating energy from the breaking down of sugars is generally considered a mitochondrial process. What's more, they found that although the PAT and ACK-controlled pathways are indeed crucial to generating energy under these conditions, and to producing an important metabolite called acetate, there appear to be other undiscovered biochemical pathways that are participating in this process as well, and capable of picking up at least some of the slack in the system. Grossman added that the system needs more work, especially if scientists are going to understand the ways in which the day-night cycle and environmental oxygen levels impact the productivity of photosynthetic organisms on our planet. This image shows localization of ACK and PAT venous fusion proteins in Chlamydomonas. The picture is courtesy of Wing-Kang Yang. With the independence for nimble pursuit, Carnegie scientists tackle some of the most profound challenges in modern science, fundamentally changing what's possible for us, our planet, and our universe.